Good morning, everyone. I am Jyoti Sharma from Indian Institute of Technology, Jodhpur, India. Today, I am going to present my work on Mendelian randomization (MR) pipeline to investigate causality between LDL cholesterol and the coronary artery disease (CAD). Before presenting my work, I would like to briefly introduce some of the topic related to MR study, the basic GWAS. As you all know, GWAS tries to find, the genome-wide association studies tries to find SNPs which are associated with the phenotype, which are associated with the phenotype using basic statistical method. These are mostly results are dependent on the p-value thresholding, as you can see here. If any SNP is crossing the p-value threshold, it is probable that SNP is associated with the phenotype modification. As we know, this GWAS can be studied and understood through the peeling tool set. It has great documentation, but not always we can see that associated SNPs are also the causal SNPs for that particular disease. It means the association and the causality is two different concepts. In epidemiological studies, we know that they try to find the determinant which are probable for any disease occurrence. The epidemiological study do this with the exposure and the outcome data sets. Exposure, we can say like any trait, or like in my case, cholesterol, their lifestyles, or any medication if they are in any. And the outcome can say that due to exposure, if there is any disease or not, like heart disease or T2D and something. So epidemiological study do this with the RCT, the randomized control trial. In RCTs, they pick voluntarily samples and they allocated randomly the two groups. One is control group and the second is the active group. For active group, they try to give some medication to them and in a restricted environment. And over the period of the time, they check the outcomes and differentiate, okay, what is the difference between the control group and the active group. There are a few disadvantages of this RCT studies. The first and main is the ethical clearance, because the sample has to be allocated in the active group and they has to give some medicine and something. And the second problem is with the biasness. If any population, if any individual have the history of diabetes, suppose family history of diabetes, even if that individual is in active group and it is going through some medication, that individual will have the probability of having diabetes more than the individual which are in the control group. It means not only the environment or the medication is responsible for occurring any disease, it is also the genetic composition, which we call the heritability. The genetic composition lead to the phenotype despite the environment and the lifestyle. So the concept of RCT is nice to incorporate the genetic information in the RCT concept. Comes with the Mendelian randomization, the MR project. In MR, the first thing, they don't voluntarily pick the sample or something. They work on the population, and in population, they check again one sample, one part of the population is studied for the exposure only, and the second part will be studies for the disease only. And after that, the common effective alleles are considered to study if there are any common SNPs or genetic composition which are both in the exposure and in the outcome, and they are any way related, correlated, and causality is there. So the main thing with MR is we have to consider a few assumptions during the study. The first, the SNP. Here we introduce it with the genetic instrument name. The SNP should be related with the outcome which we can find through the GWAS via exposure only. So that we can say, okay, because of the cholesterol, the exposure here is leading to the any disease, T2D or CAD or something. So it should not directly related to the outcome. It may be cause the multidirectional pleiotropy, which is a biasness in the MR study. We need only those SNPs which are affecting exposure and then outcome, the directionality for the precise SNP identification and the precision medicine for the further studies. And also these SNP should not be affected by any confounding factors, like we are not doing any pedigree studies. So if any disease are in their family or any related samples are in the cohort, we should exclude those for getting the generalized SNPs in the population. For data collection in such cases can be done in two ways, single sample MR and the two sample MR. In single sample MR, if some research group is studying the population, the same individual will be picked for the cholesterol studies as well as for the CAD, coronary heart diseases, the same individual. But it is not feasible all time that any research group has so much fund to genotype the all kind of data. So there is the second concept, the two sample MR. In two sample MR, in one geographical population, suppose one research group studied the cholesterol level in a sample, and then other research group in the same geographical location studied the disease. So geographical position should be the same, sample can be different, and this is called the two sample MR. My presentation focused on the single sample MR. There are advantages and disadvantages of both MR studies. In single sample MR, we will get more related SNPs to the exposure and the outcome than the two sample MR, but they are not so much generalized in terms of the population. So if we want to study the whole population in a general term, then two sample MR is a nice option. The pipeline I have presented here, I tried that with one single sample MR as well as the two sample MR data also. So starting with the method, for single sample MR, you will get the genotyping data. This is how genotyping data look like. Many of you already knew about it. We will get whole information about the sample, their SNP locations, and what are the effective and alternative allele, and the phenotype. Phenotype, if we are having disease, then it will be binary 0 and 1. And if we are having continuous phenotype like cholesterol, BMI, the continuous value will be there. As I told you, GWAS will do the statistical analysis, basic statistical analysis for the association test. So according to the phenotype, it is happening. If we are having binary phenotype, then it will go for the logistic regression. Otherwise, it will be linear regression. My co-author Vaishnavi prepared the summary statistics data from the genotype data. From genotype data, the first thing in basic quality control was performed. Then we got the summary statistics data. This is how it looked like. You can see the SNP, beta. Beta, you know, it in general term, it is how much an SNP is affecting the phenotype. The standard error, p-value, effect allele, other allele, it's fine. MAF, and the sample size. Here the sample size is different from the sample we have picked from the population. My sample size, the population size of my study was the 1401, but after the QC, it will become the 1277. This is the effective sample size. We should consider that only. And here also you can see that same SNPs is having the different beta values in the exposure, in the outcome. Exposure is cholesterol and outcome is the CAD as already written. So we try to study the impact of these SNPs, how they are different and correlated in the both data set. The summary statistics data can be found in GWAS catalog freely, GWAS catalog or MR base or something. So this is the pipeline we have prepared so far. So after preparing summary statistics data for both cholesterol and the CAD, we again do the LD, linkage disequilibrium, to check if there are any proxy SNPs with respect to that superpopulation. After doing the LD, as I told, there is no, okay, after doing the LDs, we perform the feature selection method. So for feature selection method, we should just check, okay, all assumptions are meeting or not, that all SNPs which are associated with outcome through exposure are only considered and other SNPs are removed. Then we do the feature selection. The feature selection means selecting those genetic instruments which are valid and effective for both cholesterol and CAD. We do this with the t-statistics. I will explain in the next slide. And we, again, the next step is to harmonize the data, means selecting only those data which are present both in the cholesterol and the CAD. And after that, we perform the sensitivity test. Sensitivity test is basically to check if there are any horizontal pleiotropy or multidirectional pleiotropy or not, because they will give any biasness in the result. We remove all those SNPs, and then the remaining SNPs are going perform the MR, Mendelian randomization method, already established Mendelian randomization methods. So about the feature selection method, in classical GWAS also and MR, they are all depend on the p-values, and we all know that p-value always not guarantee the significant associated or causal SNPs. So whatever data we have, like we have only beta and beta standard error other than p-value, we only have these two information. As we know, beta is the impact, how much impact it is giving, and the standard error we should have the minimum. So the t-statistics is applied on the data after the LD pruning, beta upon standard error, maximum beta and minimum standard error SNPs will be considered, and we put threshold as if any SNP is having t-statistics greater than the median, mean t-statistic, only those SNPs will be selected. This is the main feature selection method. Then there is a concept of if their selected SNP is validated or not, or it is so strong or not for that study. So F statistics is the concept introduced in one of the MR paper. They said that if F statistics is greater than the 10, then that SNP will be considered as a valid genetic instrument. But it is only applicable for the data set having size more than 5,000 to 8,000 sample. Due to the less sample size, the F statistics can be considered as 3.95. So after applying all those feature selection and MR method, these are the results. At the end of the sensitivity test and for MR, we have got the 25 SNPs, and these are the values for them. The p-value is quite nice, and these are the scatter plot for the SNP affecting LDL and the CAD, and we can see, okay, the direction is in positive. If we have a higher amount of SNPs that are affecting LDL cholesterol, that person will have higher chance of having the CAD. Other than that, we also did the causality estimation This is the causality estimation of the proposed pipeline, and this is with the default pipeline. Default pipeline means only the p-value thresholding and no sensitivity test. As you can see, there is one outlier also, and all values are near 0. But in our proposed pipeline, there are some effect. These are the overall effect, and these are the per SNP's effect. That can be studied further with the biological test. I also compare the p-value with the default method and the proposed pipeline. In single sample MR, the sample size is less. That's why the MR is not showing the significant results. Though we know that LDL is responsible, may be responsible for many heart diseases, still it is not showing any significant results. But here, despite the low sample size, the proposed method is able to detect the significant in the causality. So if I conclude this framework for effectively elucidating a causal association between exposure or outcome within the superpopulation, and the T statistics also displayed a nice result if we consider the small sample also. The sensitivity analysis in the proposed pipeline minimizes susceptibility of horizontal pleiotropy, as we see in the default casual in one of the graph. And the pipeline also extended to the 2 sample MR studies also, and we got the same kind of results. The co-author, I would like to thank Vaishnavi and my supervisor, Dr. Pankaj Yadav. Thank you.
Thank you for the very interesting talk. Now I'd like to open up to the floor for any questions.
Hi, thanks for the talk. I'm Jun from Academy of Sinica, Taiwan, and my lab is also doing causal inference, so I'm very sure to see someone doing similar work. And I'm just wondering, in your slides, are comparing MR method, right? And it seems like your p-value is like far from others' results, right? You're like to minus 23, the scale of the p-values. When you're comparing your MR method with other MR method.
Yes, that was the overall MR method p-value.
Yeah. It's like you have your method you are using IPW. Yes.
Sorry, your voice is not clear.
Sorry. Can you go back to the slide comparing like lots of MR method?
Oh, sorry.
Yeah, this one. And so you say that the p-value is great. So it's like your method is you're using IBW for reweighting your estimates, right? But other method, I'm not sure how other methods working, but it seems like other methods are also like there are MR methods. Of course, they are causal demanding method, right? And so why like your method can like to get such like significant p-values than others? What's the main difference between your method and other MR methods?
Generally, if we don't have proper information about the sample, then there are chances of the having biasness. Maybe that location is having already some ingredient that is leading to the cholesterol. So to decrease this biasness, IVW is highlighted that it will decrease all the biasness and the horizontal pleiotropy. That's why I have highlighted IVW. Okay, IVW is showing maximum result despite having, if there is any biasness, even then also it is showing the significant result than the mode and median methods. And here are the MR eager results also. It is also significant because it is lesser than 0.05. MR eager is specifically designed for horizontal pleiotropy. Okay. If by chance any SNPs is related to any other phenotype also, that is the horizontal pleiotropy, that one SNP is associated with more than one phenotype. So if there is chance that this SNP is associated with any other phenotype, the chances of this SNPs of involving in the disease is higher.
Okay, so you mean that the main difference between your method and other method is when you're considering, when you're excluding SNPs, they may have pleiotropy effect with others, right?
Right, right.
Okay, thank you.